today is start with the cumulative portion and then switch over to the non-cumulative portion to think about the different topics. And I did send this to you yesterday, so hopefully you saw that come through either on Educat or through your email. And so basically these topics were chosen based on the stats from the exams, the first three exams, and looking at areas that where there's a little bit of stumbling going on, where the stats were lower than what I wanted. And so then I would choose those topics to have you brush up on those. So having said that, if you think back to some of the other exams, some of these were essay questions and pretty detailed. For the cumulative final, I don't do that for the cumulative stuff. It's going to be kind of zoomed out, more of the big picture concepts, and I may actually not even have them as an essay question. It may be more multiple choice or drag and drop or something like that. So generally, it's not as detailed as what you think, and especially when I tend to save the cumulative, I tend to save the essay portion of the final exam for new content. All right, so then by default, that usually means if I make the cumulative questions into drag and drop or multiple choice, they're not going to be as detailed, right? I mean, you just can't get that detailed on those types of questions. So hopefully that makes sense. So the first topic is water potential. Knowing the water potential equation, just being able to write that out, and doing that correctly, that's important. And then we were looking at how each portion of the water potential equation, you have to think about that and what's going on with each one. So just knowing the basic idea of, the equation, and then what is SISA P referring to? What is SISA S referring to? And thinking about, we talked a lot about how solutes, SISA S are the solutes that are in the water system that you're looking at. And, more solutes, more negative SISA S. And so we looked at this idea, and then based on the laws of osmosis, you do get water movement if there's a difference in the overall water potential values on either side of that membrane. So remember, osmosis, it's a selectively water movement across a selectively permeable membrane. So that's what we're thinking about when we are working through water potential ideas. And talking about how the water potential the overall water potential value is definitely, you have to look at both SISA P and SISA S. And so the minute you have a solute dissolved in water, you automatically have a SISA S. And it's going to be negative because anytime you put a solute in, that's increasing the chaos or the entropy of the system, and it's denoted by a negative sign. All right. And then SISA P, especially if you have an intact cell with an intact cell membrane, cell wall, if water does rush into that, you'll definitely get a SISA P value because water will come in, and if enough water comes in, it starts pressing against the cell membrane and cell wall, and you definitely get a Turgor pressure. So just understanding those basic concepts. So if I give you a scenario and say, if water moves into this, what's going to happen? What's going to happen to the SISA S? What's going to happen to the SISA P? That type of thing. And so it won't be something where you type a big long essay. It'll be like a short scenario and just tell me based on some choices here, what's going to happen. So I don't know, do you have questions on this idea of water potential? I guess that's probably the so going through some of those slides and just refreshing your memory. The water moves from less negative water potential to more negative water potential. kind of going into, sticking with the idea of water, we talked about transpiration. And so again, this won't be a super detailed thing, but some of the major points do need to brush up on. So we did, talk about the idea that if the stomata are closed, you don't have transpiration going on. You gotta have those stomata open. So definitely the stomata really, it's all hinges on that. Is it open? Are they open or closed? And then let's say they do open up, and then that automatically will kick in a series of events that can be very good for the plant and allowing for that water movement. So when we think about movement of water through the vascular tissue, we're zooming into the xylem, so knowing that, making sure you understand what part of the body are we thinking about. And then this idea of generating a tension force. And so we got a little bit into that with the idea that the radius of the curvature of the meniscus is what we're thinking about here. So the air-water interface, you have that surface getting smaller and smaller radius of the curvature. And if you plug that into this equation, it definitely will impact your psi sub p and give you a negative psi sub p. Well, negative psi sub p, now you're talking about tension. You have a negative psi sub p, negative pressure equals tension. Okay, negative pressure equals tension. So we're looking at a tension force on that column of water in the xylem. And really, if you think about it, it's all happening up, it generates up in the leaf, right? It's the stomata, it's their doing. It's whether or not they're going to be open. And if they open up, boom, you automatically get water vapor exiting the leaf. It's not like the plant could say, oh no, vapor, you stay inside, water stay inside. It just automatically starts coming out. And as you do that, as that process is going on, you get that water evaporating off the surface of the cells inside the leaf. And you can start to imagine how it will recede into tighter spaces, which makes the curvature, the radius smaller and smaller and smaller. And so you get that nice tension force again. And so it's all starting up in the leaves. That's where it kicks off. And then it's literally that column of water is getting pulled up through this process. You get nice tension force in the leaves, less tension further away. So you get that nice gradient. And that's allowing for the column of water to get pulled up. But transpiration. Any questions on transpiration? What else did we see on that? Ah. Okay. Yeah, well, that pretty much covers it. Phloem translocation is the next idea. What forces are involved in that process? How do they get created? And then when I was going over the stats on the exam question for that, whoops, not that. I wanted you to make sure that you thought about what's going on at the sink as well as what's going on at the source. So we talked about, sugar is going through the phloem tissue, so the sieve tube elements and companion cell area. And so the sieve tube elements in particular, that's where the sugar is flowing, dissolved in the water. But now instead of a tension gradient, it's a pressure gradient. So even though the xylem and phloem are right next to each other, we have two very different ideas there. And the process is the source versus sink idea. So just making sure you remember the source sink definitions of being a source is a net exporter, so they got plenty of sugar to go around. That's typically a mature leaf. Sink, on the other hand, is probably something like roots or something that's very young and not quite up to full gear yet of being able to make their own sugars. Typically, it's good to think about roots, or in the exam, I asked about a potato tuber. So you have to think about both what's happening at the source and the sink, not just the source, in order to fully understand what's going on with this process. And so we went over the source idea let's think about the leaves and how all the cells in the leaves are busy doing photosynthesis, making lots of sugars. So you have to imagine a lot of source cells surrounding this vein, the sieve tube element, and just dumping all the sugars in there from different source cells. And so you get that nice buildup in sugars in the sieve tube element. All right. So whoops. So they're showing this buildup with the red balls there representing the sucrose. And so you have solutes getting dumped in there. More solutes, more negative psi sub S, which means that's going to drive your overall water potential more negative. And so in response, water is going to start to flow in from less negative to more negative water potential. And as soon as you get enough of that happening, that gets your psi sub P building up, building up. And so you get a nice psi sub P value. And so you get a positive pressure up in the, in the source. But that's half the story. You have to think about the other half, which is the sink. And in order to get a gradient to occur, you have to have unloading of sucrose out of the sieve tube element at the sink. Otherwise, you won't have flow from source to sink. You got to get that to happen. So if, let's say it's that potato tuber as the sink cell here, you get the tuber saying, I need all that sugar, give me the sugar. So you see all that sugar getting unloaded out of the sieve tube element. Well, now your psi sub S value isn't quite so negative anymore. It starts to approach zero. You're going to get loss of water out of there because you're going to see that it's the water will move from less negative from the sieve tube element into the xylem, which is more negative. And so down at the sink, if you're thinking about a potato tuber underground, now you have a lower pressure in the sink area. Now that's the full story. If you can say, yep, at the source we have loading, which is going to drive water in ultimately, get your pressure up. But at the sink, you have the unloading, which is going to drop your pressure. And then you have that movement of sugar from source to sink, and it's all based on a pressure gradient. But you have to have both loading and unloading going. And that's how the plant decides, if that potato tuber is all full, like, yeah, I'm done growing, I'm good, then you won't get that unloading. That signals back to the plant, oh, okay, I guess I don't have to send sugars there, I'll send it somewhere else. All right, so you have to have that communication between source and sink so the plant understands where to put its sugars throughout the growing season. So transpiration through the xylem, tension gradient, starting up in the leaves. Here with the sugar and phloem translocation, it's source to sink, and it's a positive pressure moving the sugar from source to sink. So different gradients to think about. Questions on translocation? So again, this will not be a big essay question. It's going to be some other format where you'll have to work through kind of these big picture thoughts. Okay, let's go back to the outline. Okay, biological nitrogen fixation. All right, so with that, we need to think about the okay, so I'm going to zoom out on this too. It's not going to be nearly as detailed, but some very key concepts I want you to think about is, of course, the equation, which I gave you kind of a short equation in there and then a long equation. Here's the long equation. And basically with this, really what you need to know is you're working with dinitrogen from the atmosphere, but you need a big input of energy. So knowing 16 ATP, I would still like you to know that, please. And then, so for every one dinitrogen, you need 16 ATP, and that will get you the product of ammonia, NH3. So that's what I want you to know from that equation. So again, it does require a sacrifice of energy, but it's worth it in the long run. Why is that not? It's telling me I'm not sharing my screen when I am. So annoying. All right. And then some of the other big picture ideas that were missed on the first run is the idea that in order for the plant and the bacteria to find each other, you have to go through some kind of communication, right? They have to be able to find each other in that habitat. And so knowing that there is this level of communication back and forth that allows for the microbe to find its plant partner. And so I would like you to know in terms of where did it go? Uh, the wrong one. No. Why am I not seeing it? Oh, there it is. All right. So I would like you to know that whatever chemical the bacteria excretes, we call them nodulation factors. Okay, they've come up with a group term for that. So nod factors are nodulation factors. So that's what the bacteria are excreting. As far as what the plants are excreting for communication, it varies. So, I listed out homoserine amino acids or flavonoids, but there's more to it than that. But realizing the roots are excreting something to attract the bacteria, once they get to the roots, they have to communicate back to the plant, hey, I'm a good guy, let me in. Okay, so nodulation factors serve that purpose. It's not, it's kind of analogous to you just wouldn't let anyone off the street walk into your house. There needs to be this introduction and communication before you let someone into your house. And so same idea. So that I saw was missed quite a bit on the first exam. So think about that. And then the infection thread term was missed by some too. And so just understanding that's just talking about the idea that the bacteria, once they just gently break down the cell wall a little bit, they're going to then push against the cell membrane and kind of invaginate it down the root hair. And that they're not breaching that membrane at all. They're just kind of pushing it ahead of them. And then that membrane kind of wraps around the bacteria as they're moving down the root hair. And so the whole time that this is happening, they don't have any access directly to the cytoplasm. They're always surrounded by the membrane, which is that black tube that you see. Okay, that's the invaginated cell membrane. And then when they get into the root tissue proper, they're just still surrounded by membrane material. So they're always compartmentalized. So understanding that concept too. But once the bacteria get there, that's the nodule that you start to see forming on the roots, and they're all packed in there, and then they start doing the nitrogen fixation. So just clear up any discrepancies you have about that infection thread idea and what's going on there. But that was I'm not going to ask about like dinitrogenase or like hemoglobin. Most of you did okay with that. Questions on biological nitrogen fixation? Okay, Calvin cycle. What are the terms for the three phases? What are the overall goals? And where is energy needed? So now with this, so when I say what are the names of three phases, I'm going with the pie chart. So carboxylation, reduction, regeneration. So here, knowing the three terms, and then at which point do we start to see the light reactions products are needed? So you can see the reduction phase, you have both ATP and NADPH needed. Regeneration phase, only ATP is needed. So think about this pie chart. And then what are the overall goals? Well, actually, there's a couple when you think about it in a way, because you definitely, it's a cycle. So what that means is you definitely do need to keep up making your RUBP. So that's a huge goal. They want to keep that going all day long so that when they take in their CO2, it's not wasted. They can immediately react it with RUBP. The other idea is to make sure that they are getting enough G3P so that they can make their sugars. All right. So they want to, this Calvin cycle is, directly linked into sucrose production. But in order to do that, you have to have like that enough of a pool of your G3P so that gets siphoned into sucrose synthesis, that whole process. And remember now, this is all happening in the chloroplast. I want you to know, remember, this is chloroplast still until you get to sucrose. If the plant says, you know what, I need to make more sucrose, they'll get that out into the cytoplasm and start making it there. But I'm not going to get into the step-by-step details. By the way, the regeneration question I asked on the last exam, that was the numbers game where all you had to do was show how you link G3Ps together to get your 5 carbon molecules. But I'm not going to ask that on the final. Questions on the C3 cycle? C3, let's see, what else did I say there? Yeah, so we covered that. C4 and CAM, when we look at that, what's the overall goal of the C4 and CAM? Okay, so that was a question on the last exam. At the physiological level, what do these cycles accomplish that is of particular significance? for these plants to be able to live in their respective habitats. So this is a big picture question or idea. So we go to, if you look at C4, so this is the C4. So when you look at why a plant developed the C4, it's really, it comes down to Rubisco being a sloppy, non-picky enzyme. All right. So knowing that Rubisco is kind of the weak link here. So keep in mind, Rubisco is the enzyme and it's sloppy. It can take in CO2 or oxygen. And some of these plants that live in droughty areas, warm, consistently warm, drought-prone areas, that would create a situation. If they just ran the C3, Rubisco would start to see a lot of oxygen after a while because the plant would need to close down its stomata to deal with the drought. So no CO2 is coming in, but there's still oxygen there. So now it's tipped where Rubisco sees a bunch of oxygen. So for long stretches of time, these drought prone areas, if they just run the C3, Rubisco was going to take too much oxygen in. And then you have that dreaded PCO cycle. So if Rubisco can be bathed and just surrounded by a lot of CO2, create a situation where Rubisco cannot see oxygen, then that's a pretty sweet situation. And that's what the C4 and CAM cycles do. It's not directly shown here in big flagrant capital letters, but basically right here is what you see is the payoff. You have malate coming over as a vector for CO2 that gets cleaved off malate and fed into the C3 cycle where Rubisco is. So because this initial enzyme, there it is, pepcase, because that initial enzyme could care less about oxygen, you have that situation. That's great because now CO2 is definitely going to get grabbed and reacted so that it can form into malate, and then it can easily get cleaved off of malate and just bathe that Rubisco with a bunch of CO2. So really it's that addition of pepcase that's sweet because it's so picky. It's only going to take CO2. And then that creates a situation where Rubisco sees a bunch of carbon dioxide. So very little oxygen, so the PCO won't run. And so that's what I was looking for, that you understood that this whole thing is to try to minimize PCO, make sure Rubisco sees carbon dioxide for both the C4 and the CAM, even though they're different, how they operate is a little different, but there's a lot of the same players. So really quite marvelous adaptations, depending on if they're desert plants where they've really got to worry about drought, long-term drought, high heat, or the C4 plants that are more tropical, subtropical. So again, I'm not going to ask to the level of detail that I did in terms of the pathways, but understanding the big picture ideas of why bother doing the C4 and CAM, what's it really doing at the physiological level. See if I can. Okay, and then on the last exam, there was a question on linear photophosphorylation and the Q cycle. So let me pull up. Okay. So the way to think about this. So linear photophosphorylation. Linear is referring to the idea that we're now thinking about the linear flow of electrons from starting at water, working all the way down to NADP plus. And then the term photophosphorylation, photo means light energy is being used. And then phosphorylation, start thinking about ADP, because that's really what we care about, whether you're a plant or an animal. You want to get your ADP phosphorylated so you can make ATP, right? So phosphorylation is just referring to the idea that something is getting phosphorylated. In this case, it's ADP so that you can get your ATP. So if you look at the light reactions, we went through that in detail, how you have that electron flow going all the way from water to NADP plus. And as that's happening, you get, it's through a negative delta G reaction steps, all these steps. And so you get a little bit of free energy at key points that allow for protons to get pumped essentially from the stroma to the lumen. And then what that does is it gets you your proton gradient across that thylakoid membrane. And that's what's going to get you your ATP. So when I was asking that on the last one, I wanted you to think about, okay, we know that the light reactions can make your NADPH, great. But the other key thing is ATP. And there's actually several things that lead into that, including the Q cycle. So when you look at the linear flow of electron, starting with water and then going all the way to NADP plus, okay, you can see that there is transport of protons from stroma to the lumen. And the Q cycle plays a big part of that, because as you have the Q cycle happening with the PQ coming in, excuse me, and where did it go? So here's the Q cycle. With the Q cycle, you have that PQ donating off 2 electrons. One goes linear, but the other one goes into the Q cycle. So if you look at what's going on with the Q cycle, that PQ in the cytochrome B6F needs 2 electrons. And as they do that, they're going to grab 2 protons from the stroma. So the Q cycle is, you see a disappearance of protons from the stroma because they get reacted to PQ. And then the PQ coming from photosystem 2 is also dumping protons into the lumen. With each donation of electrons, the protons get dumped. So that's happening over and over and over. And then the PQ in the Q cycle is grabbing protons from the stroma and reacting it. So you start to get this gradient. And then you can't forget too that water is also getting split, or electrons are getting extracted, and that also builds up protons. So it's really water donating protons through the electrons getting stripped, Q cycle grabbing protons, that PQ is grabbing protons, and the PQ, that's the mobile carrier, is dumping protons. And so you get this gradient where that's going to create a situation where the protons want to diffuse back, chemically speaking, and they have to go through the ATP synthase. And there's your phosphorylation event right there. So I'm not going to ask it to the same level of detail. It won't be a big essay question, but the key points on this, that when you look at the light reactions, a lot of people focus in on NADP plus getting made, or getting reduced to NADPH, which is great, but don't forget that you have this whole process linked with it that allows for ATP formation. Most of you did really well on the linear path on the question on that, so I'm not going to ask about that again. You did well on that. So it's really just that, how do they get their ATP through this process? Questions on this one? Okay. Well, then the other idea is the non-cumulative stuff, which starts with signals from sunlight. Now, the non-cumulative stuff will be more detailed in the questions, and that's where you'll see the essay questions getting derived from that content. So signals from sunlight. Basically, that's looking at photoreceptors. There's a few different photoreceptors that we covered and how they operate in order to detect sunlight and then start using that information appropriately, hopefully. So we looked at like phototropism ideas. So phytochrome, that one's a little historically that it's a tough concept. And so that's where, so the P stands for the phytochrome pigment, and then the subscripts are for what quality of light, red or far red. And it's just this weird group of pigments that toggles back and forth. And it just really does depend what quality of light that it's absorbing. It will, if it absorbs red light, then that phytochrome pigment actually physically shifts into the so-called far red form. And that's what they call the active form. What they mean by that is it's usually the one that's going to be interacting at the level of DNA. So here they show far red, kind of looking like the letter M, when it's in that state. The phytochrome absorbs red light, it actually shifts its shape, and then it moves into the nucleus and alters gene expression. But if you take the phytochrome that's in the far red form and you hit it with far red light, then it shifts back into the P sub R form. So it goes from the M shape to the Y shape, back and forth, depending on the quality of light. And so it's really, the plant is assessing both. It's looking at the ratio. How much of the P sub R do I have? How much of the P sub FR? And that helps them determine how to behave under that certain environmental situation. And so they're really interested in this with regard to how does it control flowering of plants? Because of course, we're all very interested in the flowering of plants because that's what leads to the fruit production, or, medicinal plants. So the research on this is very intense still. They have more to learn for sure, but knowing the basics of what qualities of light and that it's just this, it's a shift in the shape of the pigment that's causing this toggling back between the P sub R or the P sub FR. And then we looked at blue light, some options there with cryptochrome and phototropins, and then the UV that they can even perceive that and respond appropriately. But basically all of them, there's this change in their shape. So it's quite an elegant, simple mechanism, really. Just change your shape and that helps send the signal appropriately. So knowing, what some of these pigments are doing, how they work in terms of shape shifting. We went through that. Seems like quite a while ago. And then we got into, okay, now that we understand a little bit about photoreceptors, now we start thinking about how do plants in general perceive their surrounding and then start getting a shift in their behavior if necessary in order to adapt to that surrounding situation. So it can be this whole process of signal transduction, which can happen very quickly or somewhat slowly. There can be growth responses, or it can be like the Venus flytrap. It can be a very quick movement of the plant, but a lot of it's a growth response. So basically, the signal transduction is this flow chart here is a good one to study, looking at the idea of it's this step-by-step idea where, you know, the plant is constantly assessing its environment. So there's something going on in its environment that it's, you know, it's assessing at all. And then if it's like, oh, I better, you know, figure this out, you have a receptor in the process where typically it's often a hormone maybe that gets docked in there, and then you often have second messengers to help ramp up the signal, like, hey, really pay attention to this, let's get going, let's shift our behavior. And that helps get the signal transmission in the cell and tissues. And ultimately, it's usually down to the level of DNA, where you have to get some shift in the DNA, some gene expression going in order for the plant to respond appropriately to whatever it's dealing with in its environment. And so the big picture idea here, and then we went through and looked at, you know, it's often, at first they thought all the receptors were on the cell membrane, and then they realized with further careful study, not really. Sometimes you have receptors inside the cell, but you have to have a receptor somewhere where there's some kind of chemical signal. And then the second messengers just kind of help ramp up the signal and really make sure that it continues all the way to completion. So they're finding calcium's a huge player in that, but there's others too. Something as simple as pH. I mentioned a little bit about the lipids. Sometimes those act as signals, like if a pathogen's chewing away at the cell membrane and some of the lipids break free, that can be a second messenger actually. Reactive oxygen species, we talked quite a bit about that too in this section, as well as the plant defense. So this is just showing a lot of calcium, pH shifts going on. Not really showing the damaged limb membranes, but you can get that. And then the reactive oxygen species. So that's actually a much bigger player than they thought in terms of all of these environmental situations can trigger reactive oxygen species as second messengers. And then we looked at hormones a little bit too in this slide set and started talking about how they're important signal molecules. So we kind of went through all the big fives with auxins and cytokinins, gibberellins. So knowing what they are, you know, auxins very famous for being produced in the tips of stems, especially, and that's what's, you know, very famous for the phototropism response, getting that bending toward the unidirectional light. So knowing what the 5 hormones are, a little bit about what they do for the plant. And then we didn't get too much into detail toward the end here. So we looked at signaling pathways, like how is that actually done? I would like you to know that it's based often on the 2-component system. So that's kind of a big picture idea. They kind of stole that from bacteria, but very often they'll run some kind of signal transduction system like that, where you have a sensor that's sensing the environment, and then the response regulator, and you see a transfer of phosphate in order to relay the signal. All right, so that's a pretty important concept that goes for plants, goes for you, goes for microbes, a bunch of animals. So this is pretty universal. So that would be a good thing to grab onto. But then it gets very complicated pretty quickly. So we're not getting into the details of these last set of slides at the end of this slide set. I was just showing them to, you know, bring home the point that you get the docking into the receptor protein and the shift of the phosphate from sensor to the response regulator. And then the other thing to think about is this slide where I have in the title here, it says figure 12.19, signal transduction pathways in plants often function by inactivating repressor proteins. That's important. I don't know, that may not sound important to you, but it is. So basically it's the idea that there's a repressor protein on a section of DNA by a gene, and it's just stopping expression. And then if the plant's like, I need that up and running, it will create a situation where those proteins come off. They're often degraded and recycled. And so essentially they come off that piece of DNA, and now you can get it to get transcribed and expressed. So that's a pretty common strategy with plants. So inactivating repressor proteins in order to do their gene expression. All right, signal transductions. What else did we do? Signal. We did. What's the next one? Oh, plant defense. So then plant defense, the way I think would be the best to study this would be mechanism of defense. So when we were looking at this, it's kind of broken down into how does a plant deal with some kind of herbivore versus how does it deal with a microbe pathogen? Don't forget about just some of the general defenses that they talk about in this section. So the chapter reading in the book backs up a lot, pretty much everything we say in the slides here. But when you look at plant pathogens, herbivores, pests, we talked a little bit about them, like different ways of attacking, being herbivorous, whether they're phloem feeders, cell content chewing, please know the difference between those. But then we started looking at, okay, if an herbivore comes along, what does it do? So that's getting at, here it's showing slide 17, working your way forward where they're showing herbivory going on. And so the way they respond to herbivores is a little different than how they do it with microbial pathogens. And so here we were looking at, and then there's the terminology. So elicitors, you need to know what the terminology means. That's coming from something from the herbivore, the whatever's attacking. And so then they need to figure out, okay, what's attacking me? So they pay attention to those elicitors, and they can be all kinds of different things. Maybe it's something in the saliva of the caterpillar. Maybe it's something coming off the body of something chewing on it. And then you often see a huge upswing in calcium as a second messenger. Hormones kick in. And then this idea that you do have certain hormones and they differ. It depends on if it's an herbivore attacking or a microbe. So there's different hormones, which makes sense because you have to have different signal transduction pathways that the plant can tell the difference between. So it knows, oh yes, this is an herbivore. Oh no, it's a fungus. So think about those. So this slide set is kind of, we talk about herbivore attack first, and then further down, when I say pathogen, that's getting at microbes. So the pathogen infection content, plant pathogens. And so they work it a little different. And so that's one thing I've noticed in previous classes is kind of, if you don't separate that when you're studying, it tends to get muddled and crosses, and then it's not a good situation. So kind of break that down as you're studying into the two separate herbivores, microbes. And there's different hormones involved, slightly different things. And then with pathogens, we did look at, quite a bit of, they have options. So they, in this white box here showing, hypersensitive response, phytoalexins, lignin biosynthesis, and then the hormone and enzymes. So thinking about different ways to attack. So historically, this slide set, I think, is the one that is most tricky for students. And the other stuff is a little bit less tricky. So please make sure that you spend some time with this. There will be essay questions on these ideas, so. And then the abiotic stress, I didn't get into huge detail on any one subject of that, but there's chilling and freezing. What else did we do? Chilling and freezing, salt, pollutants, and flooding. But there's not a whole lot of content on those. So this slide set's probably the most detailed. Okay, we're running over. So next week, there's no classes, which means my schedule is more open. If you want to meet with me to ask questions or go over anything, please send me an email. We can do that. I won't necessarily be working in my office all day. I have a very sick dog at home. I'm trying to keep an eye on her. So, but I'm happy to come in. I can meet via Zoom. I'm happy to help you, and my schedule is much more flexible next week. So if you have any questions at all, or if you just want to send me a quick, hey, can you clarify something for me? I'll be checking my email quite a bit. So please reach out to me if you have any questions, want to meet, okay? And then I'll see you here on Wednesday at noon. Duncan and I will be here, so good luck with all the rest of this.